The Wuhan Coronavirus is one of the top trending topics people are concerned about today. As you might have heard, the Huanan wholesale seafood market could have been the starting point of this major outbreak. Many people believe that this is likely a spillover incident, since the genetic sequence of this virus greatly resembles a recombination between two different coronaviruses that may have originated from snakes and bats. It is undeniable that the wildlife market offers an opportunity for close contact between animal hosts and humans. So here's a brief summary of what has happened thus far. The first case of the coronavirus happened on 12th of December. On the 1st of January, the suspected Huanan seafood market was shut down, following which on 9th of January, the coronavirus claimed its first life, a 61-year-old man. On January 13th, cases were discovered outside of China in tourists in Japan and Thailand. By the 21st of January, the virus officially reaches the US with one confirmed case in Washington state. It also appeared in Singapore as well. China has nearly 300 confirmed cases, of which 6 people have died. On the 22nd of January, China suspended all travels out of Wuhan, placing the city under quarantine. There are nearly 600 confirmed cases worldwide, 444 just in Wuhan. Preliminary research suggests that the virus may have originated in snakes, but some experts refute the finding. 17 people have died. On January 24th, China quarantines 8 more cities in the Hubei province, trapping 35 million residents in their cities. CNBC reports that the death toll has climbed to 41, and the total number of infected has risen to 1,287 people in 12 countries. All fatal cases have occurred only within China. France confirmed its first 2 cases as well, the first appearance of this coronavirus in Europe. The CDC announced on 28th of January that it recommends that travelers avoid all non-essential travel to all of China due to the virus outbreak. The Chinese government announced on 26th of January that it was banning the sale of wildlife at markets, restaurants and even online. About 35 scanners were planted at Changi Airport as precautions were stepped up following the outbreak of this virus. The Singapore government is also giving every household 4 masks after 5 million stocks sold in just 9 days. Airports in Malaysia, Canada and Thailand have implemented different measures for screening passengers who may be infected with the deadly coronavirus. Thermal screening has been used to check the temperature of passengers to detect symptoms of fever and would not hesitate to quarantine them if necessary. 1,500 personnel from the Singapore Armed Forces aim to pack 5.2 million masks in a 24 hours operation. As of 30th of January, the virus has spread to at least 19 other countries. Just to name a few, Japan has 11 cases, Singapore has 13, US has 5. Worldwide, 170 people have died, and more than 7,700 are fighting symptoms, including fever and breathing difficulties. The Wuhan virus has now infected more people in China than SARS. According to the World Health Organization, 16 to 21% of people with the virus in China became severely ill, and 2 to 3% of those infected have died. You might have known that the incubation period, that is the time required for the virus to replicate and for symptoms to be seen, is from 3 to 14 days. It might be possible for people to be infectious without having any symptoms, and that would be a concern. If someone gets sick, you want to be able to identify them and track their contacts. If someone transmits the virus but never gets sick, they may not be on the radar at all. It also makes airport screening less useful because people infectious yet do not have symptoms would not be detected. According to the CDC, the virus can and has been spreading from person to person in many parts of China. In terms of how one would catch the virus, the CDC says that they are most commonly spread via the air from viral particles from a cough or sneeze. Close personal contact like touching or shaking hands, or a surface with viral particles on it. And what are the common symptoms of coronavirus? It includes fever, dry cough, shortness of breath, and breathing difficulties at the outset, and possibly gastric issues. Severe symptoms include a fever, pneumonia, kidney failure, and in very rare cases, death. So what exactly are coronaviruses? The coronavirus is a group of zoonotic spherical-shaped viruses that can cause illnesses in both humans and animals, with severity ranging from a simple common cold to more severe diseases such as SARS that kills roughly 1 in 10 people infected. Here are some important structures of the coronavirus you should know before moving on. Coronaviruses are a family of envelope viruses covered with crown-like spike proteins on their surface. These spike proteins serve as receptor binding domains that allow for cell-to-cell recognition. And what do I mean by that? For the virus to enter into a human cell, the proteins found on its surface must have a specific 3-dimensional shape complementary to that of the cell receptors found on the surface of human cells. This will then allow for the attachment of the virus onto the human cell to facilitate its entry for viral replication. There is also a single-stranded positive strand RNA found within the envelope in the nucleocapsid, basically a protein code. Just like most genomes, it contains a 5' cap structure along with a 3' poly-A tail, allowing it to act as an mRNA for translation of the replicase polyproteins. What's more important here are the RNA structures required for replication and synthesis of viral RNA, and they are found at the 3' UTR. The organization of the coronavirus genome is a 5' site, followed by leader, UTR, replicase, S, which stands for spike, E, which stands for envelope, M, which stands for membrane, N, which stands for nucleocapsid, and lastly the 3' UTR poly-A tail. Just to be clear, viral proteins are produced from these genes found in the genome. Protein synthesis consists of 3 levels. The gene level, which is what I have just mentioned about the S, E, M and N genes, will have to undergo transcription to form mRNA, following which translation to form viral proteins before they are released out of the human cell. So the S protein has an N-terminal signal sequence to gain access to the endoplasmic reticulum since it is heavily glycosylated. The endoplasmic reticulum serves to package newly replicated viral particles at the end of the cycle. In most, but not all coronaviruses, S protein is also cleaved by a host cell protease, which is an enzyme, into 2 separate polypeptides, noted S1 and S2. S1 makes up the large receptor binding domain of the S protein, while S2 forms the stop of the spike molecule. The M protein serves as a transmembrane protein that may adopt different conformations that promote membrane curvature of the virus. The E protein facilitates assembly and release of the virus and controls ion channel activities, sometimes required for pathogenosis. Lastly, the N protein constitutes the only protein present in the nucleocapsid, which is the protein code. It serves to hold the proteins produced closely with the replicase transcriptase complex, in short RTC, and subsequently package the genome into viral particles with its binding capabilities. So having known all this, I'll put into place how the coronavirus infect and replicate in the human cell. This life cycle involves a total of 5 stages. The first stage involves the attachment and entry of the virus into the cell. As previously mentioned, the spike protein recognizes and binds to the receptor binding domain found on the human cell, and this triggers a conformational change in the spike proteins, which then mediates fusion between the viral cell and the cell membrane on the human cell. This results in the release of the nucleocapsid containing the RNA from the virus to the human cell. The RNA contains a replicase gene, and that brings us to the second stage of this cycle, called the replicase protein expression. This replicase gene codes for 2 co-terminal polyproteins, namely Rep1A and Rep1B. To produce these proteins, the gene then undergoes transcription and translation. These polyproteins contain non-structural proteins 1 to 60, and each protein has its own specific function. For instance, protein 1 serves to promote cellular mRNA degradation and blocks human cell translation, resulting in the blocking of our immune system. These 2 co-terminal polyproteins are then cleaved and separated into 16 non-structural proteins for them to exist individually, which they will then gather to form what you call a replicase transcriptase complex. And this creates an environment suitable for RNA synthesis, and ultimately are responsible for the replication and transcription of the subgenomic RNAs. Do you remember the SMEN proteins that we were talking about? Yes, those are produced via transcription and translation of the SMEN subgenomic genes with the presence of the replicase transcription complex RTC produced. This brings us to the third stage of the life cycle, where the subgenomic RNAs then replicate, transcribe and translate to form proteins. After this is done, the proteins go through the endomembrane system to repackage and chemically modify all these viral proteins that are produced. The M protein directs most of the protein-to-protein interactions required for the assembly of the coronaviruses. The last stage is the release of the virus to infect more neighboring cells. The S protein that does not get assembled into the complex transits to the cell surface, where it mediates cell-to-cell fusion between infected cells and adjacent uninfected cells. Due to the capability of the S proteins, many of such viruses can bypass the virus-specific antibodies without being detected. So what about the Wuhan virus? The coronaviruses are also known for their ability to recombine using both homologous and non-homologous recombination. The ability of these viruses to recombine is tied to the strand switching at the gene level. In fact, we are dealing with a novel coronavirus, which is a new coronavirus that has never previously been identified before. Why so? Because of genetic recombination. Researchers have used the analysis of protein codes to deduce which animal host it originated from. And the genetic sequence of the protein they have been studying about seem to suggest that it came from bats and snakes. No bats were sold there, only snakes. Snakes often hunt for bats in the wild. This raises the possibility that this coronavirus might have jumped from the host species, which are likely to be bats, to snakes, and then to humans to cause the outbreak. Due to the recombination, there is a change in one of their viral proteins that recognizes and binds to receptors on human cells. This recognition is a key step in allowing the virus to enter our cells, and the change in this specific protein may hence have contributed to its ability to hog the species. Having heard all this, die-hard pessimists may contend that the world would be wiped out by the virus. Some would even buy 100 boxes of masks and 1,000 thermometers to feel more secure. I mean, even the World Health Organization declared the ongoing corona outbreak to be a global public health emergency. Indeed, it may be inevitable that the virus will continue to spread to more people and claim more lives. However, one should not worry, the world's not going to end. We must stay calm and work responsibly together as a community. To be calm but not to let our guard down. So wash your hands frequently, use your mask properly, check your temperature regularly, avoid large crowds, and never touch your face with unclean hands. Coronavirus really isn't as serious as you perceive. We should trust our government and community that they have what it takes to minimize the impact of this outbreak to its lowest level.